Welcome to another video for biology. In this video we're going to be discussing the replication of DNA, the copying of DNA. So as a cell prepares itself to be copied and multiplied, it needs to make a copy of every single component inside the cell, including DNA. This copying of DNA happens in the cell cycle in the S phase, okay, or the synthesis phase. So this is where your DNA will copy itself. Okay, so it copies itself, it doubles itself to be shared into two new cells in the mitosis phase. Excuse the background noise. I am recording this outdoors, so you might hear cars and such going behind me. Well, I need you to keep three objectives in mind as we continue on in this video. Number one, you need to know how we go from a one-stranded DNA to from a one double-stranded DNA to two double-stranded DNAs. Okay, so again, we're making a copy of DNA. Secondly, I need you to understand how DNA replicates itself, so you need to know these two terms, lagging strand and leading strand. Okay, and then finally, I need you to recognize the roles that enzymes play in the replication of DNA. Alright, let's go ahead and just dive into this now. So we have here a double-stranded DNA molecule, as you know, is wrapped around, supercoiled, wrapped around in histones in chromosomes. So we're going to isolate our DNA molecule, and remember that DNA runs anti-parallel. So it has an anti-parallel configuration. So you're going to notice the sugar molecules, the deoxyribose sugar molecules, they're going to change position. You notice that? Remember the orange dot represents the position of the oxygen in that sugar, and you'll notice that they're pointing in opposite directions. Okay? So let's take a closer look at these sugars. Okay? So again, each strand is anti-parallel, going in different directions, right? So the top one is going towards the right, and the bottom row is on the going towards the left. Well, we want to take a closer look at these sugar molecules because we want to know how do I figure out what direction the sugar molecule is going. Okay, going left, right. How can I be more positive as to which direction they're going? Well, as we look at the sugar molecule, okay, remember that orange dot represents the position of the oxygen. At that point, I identify first my oxygen, and then I count my carbons going clockwise. So my first carbon position is right there. So you'll notice each corner in that pentagon represents a carbon position, okay? So I go clockwise, carbon 2, 3, 4, and then my fifth carbon is outside of my pentagon. Okay, this is actually very important. In fact, just to kind of make it easier on yourself, well, just understand that this particular sugar molecule's configuration is going from a 3 carbon from the bottom to a 5 carbon, so it's going in the up position. But something to keep in mind, like I was mentioning before, to make it easier on yourself, identify where that fifth carbon is easily done by looking at where the oxygen molecule is positioned, because that 5' carbon molecule, and that's what that little not a comma, but that little apostrophe there, whatever it's called in grammar, that just means prime. So this is said 5' and this is 3'. So this is the 5' position of the sugar molecule. Just look for that oxygen molecule. That oxygen molecule, think of it as an arrow pointing towards the 5' direction. And then our 3' would be opposite. Okay? So this is going from a 3' to a 5' position. So now as we look back on our previous slide, we see here that the top row, again, my oxygen molecule is pointing towards my 5'. So this row is going from 3' to 5'. And since the other strand is anti-parallel, that's going from a 5' to a 3' position. Okay, so I hope you can see how this configuration, this anti-parallel position, or looking at how the sugars are positioned, you can tell what direction the molecule, the strand is going. This is going to play an important role in understanding how DNA replicates itself. Alright, well, let's take a second to look at the different enzymes that are going to be playing a role. First of all, we have our DNA molecule. Next, we have the enzyme called helicase. Okay, and you'll notice it has a zipper, because that's responsible for unzipping the DNA double helix. You have DNA polymerase. You notice I made him into a little construction worker because, well, you'll see why I have these different little images this way. And then we have our RNA primase. It has a starter flag there. And then we have our DNA ligase. Actually, I think this might be a finishing flag, but either way, let's just call it a starter flag. And then our DNA ligase, and you see I have Elmer's glue symbol there, and you'll see why later. These are the terms that you need to know by the end of this video. Semi-conservative, Okazaki fragments, leading strand, lagging strand, and 5' to 3'. Alright, well, let's dive in. So, we need to copy DNA. DNA is bonded by hydrogen bonds. Helicase will come in, will attach itself to a specific portion of DNA. It will break these hydrogen bonds, and it will slowly, actually not slowly, but it will begin to unwind, or it will begin to unzip this DNA molecule. Alright, so let's speed this process up just a little bit. You'll notice that there's a Y shape. You'll notice there's a Y shape that begins to take form, and at that point right here in the Y, this is called the replication fork. This position right here, okay? This part, the V part of the Y shape, okay? So that's a replication fork. DNA is synthesized, always synthesizes in the 5' to 3' direction. Okay? So if I look at my top strand, that strand is going from 3' to 5'. So my 5' are on the right, and all my 3' are on the left. Right? So that means that if my parent strand is 3' to 5', that means my new strand has to be 5' to 3'. It has to be anti-parallel. So you notice the position of that sugar? It's opposite to the parent strand. Okay? So DNA polymerase, the enzyme that's going to attach DNAs to it, are going to attach all the DNA molecules in that position. That's going to be anti-parallel. And it will be copying towards the replication fork. Okay, remember the replication fork is this spot right here. So that leading strand, this is called the leading strand. This is a continuous replication. It copies, it lays down DNA nucleotides from 5' to 3' direction. 5' to 3', right? Remember, here's my 5', and then leading down at the end is my 3'. And it copies towards the replication fork. And this is a continuous replication. Okay? Okay, that's my leading strand. Alright, now let me introduce you to this enzyme right here, DNA polymerase. I know we've discussed it before, but let's now look at what it actually does. So DNA polymerase is like my construction worker who carefully puts together the appropriate DNA nucleotide to the template, which is the leading strand. This is my parent strand. So the DNA polymerase will read the parent strand, and it will notice which nucleotide needs to go, and it attaches the appropriate nucleotide. Again, this is in a 5' to 3' direction, right? Alright, now let's take a look at our lagging strand. Our lagging strand synthesizes DNA in the opposite direction. Okay, so on the parent strand we went from left to right towards the replication fork. Well, in our lagging strand we will synthesize DNA from our replication fork, well, away from our replication fork, right? Okay, well, let me introduce you now to our second enzyme, which is our primase, which is a primer. Okay, DNA ligase will come into play later. Okay, alright, let's look at what our primase does. RNA primase places RNA, it's an RNA molecule, not a DNA molecule, but an RNA molecule. Okay, and it places it in certain positions along the DNA molecule, and it serves as a starting point for our DNA polymerase. So you notice this is our lagging strand down here. Remember, we're going to end up copying away from the replication fork. So these RNA primers are starting points for our DNA primase. So our DNA primase identifies the first position, and you notice it's copying away from the replication fork. And as it approaches each RNA primer, it replaces it with the appropriate DNA nucleotide. And each one of these sections is called an Okazaki fragment. Okay, so that's one Okazaki fragment. Here's our second. And remember, our RNA primers mark the starting and end points for each Okazaki fragment. Okay, and we continue down the line. Here's our third Okazaki fragment. Okazaki fragments can be anywhere from 100 to 200 nucleotide bases long. Okay, so these are actually really long fragments. I know I've shown here like 4 or 5, but just keep in mind that's just for our simplicity's sake here. Alright, so before we move on, let's go back. Okay. Notice again that each DNA molecule is anti-parallel. So I go from the 5 direction to a 3 direction. Remember, DNA polymerase lays down DNA nucleotides in the 5' to 3' direction, whether it be the lagging strand or the leading strand. Okay? Alright, well, let's move on now to the next part. Well, on our lagging strand, this strand is still not completely stable. So what's going to happen is our DNA ligase, which are Elmer's glue, our DNA ligase is going to identify all the positions where our RNA primers were before, and it's going to go down the line and it's going to glue together, if you will, or bond these new DNA nucleotides to the rest of the DNA molecule and make this double strand more stable. Okay, so our end result is we have 2 double-stranded DNA molecules. Each double strand has a parent or an original strand, and then each strand also has a new strand, a newly copied strand. So we have two double-stranded molecules with each having one parent strand and a newly copied strand. Alright, so here again are your objectives, so you can go back and review and make sure that you understand each one of these 3 ideas. And then here we have our different enzymes that play a role in this process with the key terms that you need to know. So please make sure and review these enzymes and understand their role. Go back and look at these terms and understand what they're trying to say as well. Okay, good luck in your study.